Welcome to Efiwe, excellence without stress.
Hi, there is a few ways. Our topic today is optical instruments. Today we're going to study so many optical instruments and their applications. So the first optical instrument we're going to talk about today is the simple lens camera. So I'm sure most of us are familiar with a camera. Yes, we've seen a camera. Our phones has cameras, and we have actually seen this type of camera that is on the screen. So the simple lens camera consists of a light-proof box with a converging lens. I'm sure in the previous class we spoke about the converging and the diverging lenses. So looking at this box here, we have the converging lens in there. So the simple lens camera consists of a light-proof box with a converging lens in front and a light-sensitive film at the back. The function of the lens is to focus images of the objects to be photographed onto the light sensitive plate or film. That's the function of the lens. There is a provision for adjusting the distance between the lens and the film so that objects in front of the lens can always be sharply focused on the film. Sometimes the convex lens is replaced by a system of lenses. So the simple lens camera has a fixed distance between the lens and the film. That's the fixed distance between the lens and the film. This distance is equal to the focal length of the lens. The distance is equal to the focal length of the lens. The diaphragm regulates the size of the aperture. This is the aperture here. So the diagram regulates the size or opening, which controls the amount of light energy sensitizing the film. The shutter, this is the shutter here. The shutter of variable speed between the lens and the film controls the time interval during which light is allowed to fall on the film. This time interval is called the exposure time. The process of making a photograph begin and end with light. That's it. Making a photograph to begin and end with light. Rays of light enter a camera and are focused into an image. To take a photograph, the camera lens is pointed at the object and the focusing ring is used to adjust the distance of the lens from the film until a sharp image is seen on the film. When the button is pressed, the shutter quickly opens and closes, exposing the film for a brief period to light from the objects. This is just the principle of how the camera works. Then we have another optical instrument, which is the projector. The projector is used to project a small image and making it big on a large screen. So the projector is an instrument for projecting on a screen an enlarged image for a transparent object, e.g. a slide. The function of the various parts of the projectors are: the small but powerful light source is to illuminate the object. The light source, we can see it from this diagram here, the light source is to illuminate the object, which is usually what? Non-luminous. We've spoken about this. A converging mirror placed behind the light source helps to direct the light onto the film or slide. This is the converging mirror. The concave, is also called converging. So this is a converging mirror. So a converging mirror placed behind the light source helps to direct light onto the film or slide. The condenser, as we can see here now, these are the condensers. The condensers consisting of two plano-convex lenses collects the light from the light source, yes, which is this one, and concentrates it on the slide. This is the slide or transparency, and illuminates every part of it strongly and evenly. Between the two slides is usually an infrared filter. Between these two slides is usually an infrared filter to prevent the passage of heat to the slide, which might otherwise melt it. So the slide carrier is a framework in which the slide is placed upside down so that it will appear erect on the screen. The projector lens placed near the slide produces a real enlarged and inverted image of the slide and focuses it on the screen. The lens is at a distance of between f and 2f from the object, where f is the focal length of the lens. See, in the previous, this is the projection lens. This is the projector lens here. The screen, usually white, receives the image, which is the right way up, if the object is inserted upside down into the slider carrier. The principle of the slide projector is used in cyanoprojectors, photographic enlargers, and several other devices. So functions of each part of the eye. This is the diagram of an eye. So we have different parts. We have the choroid, the retina, the macula, the optic nerve, the optic disc, the sclera, conjunctiva, the cornea, pupil, and so on. So the major part, reason why we're showing you this is just to explain the relationship between the eye and a camera. So the cornea, which is this part, it refracts the light and protects the eye. That's the function of the cornea. The iris controls how much light enters the pupil. That's the iris. Then the lens, this is the lens. You can see the lens looking like the normal lens we saw, like the convex lens, focuses light onto the retina. So this is what the lens does. It focuses light onto the retina, which is here. The optic nerve, this is the optic nerve here. The optic nerve, the bundle of sensory neurons that carry the impulses to the brain. Then the retina, layer of tissue at the back of the eye that contains photoreceptors called rods and cones. That's the retina. This is the retina here. Then we have the fovea, area of the retina with the highest concentration of cone cells that provides sharp vision. Focus on that. It said sharp, that provides what? Sharp vision. Then we have the aqueous humor, the vitreous humor, the ciliary muscles, the sclera, and the pupil. All these are different parts of the eye, and these are their function. The pupil, hole in the center of the eye that lets light in. This is the pupil. This is the hole in the center of the eye that lights are in, and this is the sclera here, tough outer layer that extraocular muscles can attach to, and those are the parts of the eye and their function. Accommodation. The eye has an adjusting mechanism for focusing the image of an object onto the retina. It adjusts the focal length of the crystalline lens through the action of the ciliary muscles, which control the curvature and hence the focal length of the lens. Thus, through this mechanism, both near and far objects can be focused at the retina. So what is accommodation? Accommodation is the ability of the eye to alter or adjust the focal length of its lens so as to form clear images of objects at different distances on its retina. Again, accommodation is the ability of the eye to alter or adjust the focal length of its lens so as to form clear images of objects at different distances on its retina. This adjustment is brought about through the action of the ciliary muscles. So now, defects of vision defects. So we have what is called the long sight, which is hypermetropia, and the short sight, which is called myopia. We have the hypermetropia and the myopia and its correction. A long-sighted person can see far objects, just as the name implies, long, far objects, but cannot see near objects. They can see far objects but cannot see near objects. Its near point is farther away than 25 centimeters. That's the normal near point. The eyeball is too short, or the crystalline lens not sufficiently convergent. It doesn't converge. Then the defect is corrected with the aid of a suitable converging lens placed in front of the eye. So the defect is corrected with the aid of a suitable converging lens placed in front of the lens. Look at the converging lens here. So that's what we use to correct long-sightedness. You use a converging lens to correct long-sightedness. Then we have the short-sightedness. A short-sighted person cannot see distant objects clearly. This is because the eyeballs are too long, just the opposite of long-sightedness. So the eyeballs are too long, or the crystalline lens is too converging. The rays from distant objects are brought to a focus in front of the retina instead of at the retina. So correction is done with the use of a diverging lens. You can see the diverging lens here. Correction is done with the use of a diverging lens. Those are the defects of vision. So the comparison between human eye and the camera. What are the similarities? The camera includes a light box painted black inside. The human eye is impregnated with black pigments within. So also the pupil in the eye serves the same purpose as the aperture of the camera. Let's take note of that. The pupil in the eye serves the same purpose as the aperture of the camera. Both the eye and the camera have a converging lens to focus rays from an external object. Also, both have light-sensitive materials on which the rays are focused, the retina in the eye and the film in the camera. So that's it, the retina in the eye and the film in the camera. The iris in the eye serves the same function as the diaphragm in the camera. The iris in the eye serves the same function as the diaphragm in the camera to regulate the amount of light entering the equipment. So the iris regulates the amount of light entering the eyes. Also, the diaphragm does the same thing. So what are their differences? The human eye has a power of accommodation, that is the ability of the lens to focus points at different distances on the retina by altering the focal length of the lens. Hence, the eye lens has a variable focal length, but the focal length of the camera lens is fixed. That's the difference. Also, the distance between the lens and the retina is fixed in the human eye, but it is variable in the camera. That's their similarities and relationship and differences. So this is another optical instrument. So we are going to microscopes and telescope. The first microscope we're going to talk about is the simple microscope or the magnifying glass. Most of us have seen this before. It is a convex lens, you can see that, which is used to produce magnified images of small objects. It is used for reading small print and for studying biological specimens. Also, the lens is moved until the image is seen distinctly at 25 centimeters from the normal eye. The image subtends a larger visual angle than that produced when the object itself is placed 25 centimeters from the unaided eye and therefore appears bigger. So this is the microscope. This is what is called the microscope or the magnifying glass. So we talk about the compound microscope. This is the diagram of a compound microscope. So we have different lenses there. We have the eyepiece, we have the objective lens, and so on and so on. So to produce a higher magnification than that obtained from a simple microscope, a combination of two convex lenses. So don't forget that for a compound microscope, we use how many lenses? Two convex lenses is used in an arrangement called the compound microscope. The compound microscope consists of two converging lenses of short focal length. The lens nearer this object is known as the objective lens. Don't forget that. The lens nearer to the object is called the objective lens, and the lens through which the final image is known as the eyepiece. Let's take that again. The compound microscope consists of two converging lenses. It consists of two converging lenses of short focal length. The lens nearer to the object is known as the objective lens. So this is our objective lens, nearer. This is the one that is close to the object. If we want to see the object, we'll put the object here. So this one is called the objective lens. And the lens through which the final image is seen, the object through which the final image is seen, is known as the eyepiece. The eyepiece has a comparatively larger focal length than the objective lens. So this has a larger focal length than the objective lens. Since the focus of this one is higher than this, so it can focus more than this other part. So the eyepiece has a comparatively larger focal length than the objective lens. So that's for the compound microscope. So also the astronomical telescope. This is another optical instrument, the astronomical telescope. The telescope is used in viewing distant objects such as the stars and the planets. I'm sure most of us have seen this, if not in real life, but in movies. The simplest telescope is the astronomical telescope, and this is it. This astronomical telescope uses two converging lenses, as we can see, C1 and C2. It uses two converging lenses, the objective lens and the eyepiece respectively. The objective lens has a focal length, whereas the eyepiece has, the objective lens has a long focal length, while the eyepiece has a short focal length. This is for the astronomical telescope. The astronomical telescope produces an inverted image. Can you say that? It produces an inverted. We've already talked about this many times. It produces an inverted image, that is an upside down image. So that's for the astronomical telescope. Then we go to the Galilean telescope. This type of telescope uses a convex lens of long focal length as the objective and a concave lens of a short focal length as the what? Eyepiece. Look at it now. Use this as the eyepiece and use this as the objective lens. So this type of telescope uses a convex lens of long focal length as the objective lens, and uses a concave lens of short focal length as the what? Eyepiece. The two lenses are mounted so that they have a common axis. Look at them, they are mounted common axis with the distance between them equal to the difference between their focal length. The distance between them is equal to the difference between their focal length. The objective lens collects parallel rays from a distant object. This is it. It collects the rays. You can see the rays coming in. It collects the rays from a distant object to form a real and a real inverted image at the principal focus. To form a real inverted image, you can see it now, at the what? Principal focus. That's for the Galilean telescope. So we have the prism binoculars. They are essentially a pair of astronomical telescopes, each of which is fitted with two right-angled isosceles prisms. Two right-angled isosceles prisms. The light passing through the objective is reflected from one prism P to another and then emerges through the eyepiece. This is called the prism binoculars. The function of the prism is thus to really invert the image formed by the objective and present an erect image. So what it does is it's reinvert. If the image formed is inverted, it does what? It reinverts it and presents an erect image of the object to the eyepiece, which is subsequently magnifies it. An erect and magnified image is then presented to the viewer. That's what the binoculars, prism binoculars does. I'm sure we have seen this in movies. The prism binocular, as well as presenting an upright image, are also very compact since they reduce the optical part of the light ray from the objective to the eyepiece very appreciably. And that is all about the prism binoculars. So this is all about the optical instruments. We've talked about the microscope, the simple camera, the telescope. We talked about the relationship between the eye and the camera, and that is all for optical instruments. Thank you guys. See you in our next class.